Let's try to understand four pathways of pyruvate. So pyruvate is a end product of glycolysis when we convert glucose into pyruvate via these enzymes called glucokinase, hexokinase, uh, phosphofructokinase 1. This is the rate limiting enzyme of this glycolysis, and then in the end we get pyruvate kinase, and then we can convert pyruvate kinase to pyruvate. Later, pyruvate can be used into four different pathways. Let's try to understand all of these. Pyruvate can be used in four different pathways: are lactic acid, alanine, oxaloacetate, and acetyl CoA. So that's the that's the four pathways. Now, depending upon the energy demand, depending upon the type of tissue, depending upon the availability of oxygen, we can convert pyruvate into these four things. Okay. So lactic acid is usually converted, usually converted in anaerobic conditions, because this pushes, this is why we have lactic acid, because we don't have O2, right? Now, why it happens, why pyruvate turns into lactic acid, is because no oxygen means the electron transport chain cannot generate NAD+, okay? Now, this is the pathway. Pyruvate turns into lactic acid, right? And the enzyme that is used to convert pyruvate into lactic acid is lactate dehydrogenase. Now, this enzyme is activated under anaerobic, hypoxic, infection, and heart failure conditions. So think about lactic acid, think about levels of lactic acids, think about these conditions. Other thing that it can, other meaning, another like other physical meaning of this is that once the electron transport chain cannot generate NAD+, the LDH, this lactate dehydrogenase, LDH, it generates NAD+ by converting pyruvate into lactate, right? And this keeps the glycolysis running, producing small amount of ATP even without O2. So that's the physical relevance, right? Now, why it's found in RBC, why it's found in WBC, is simple that these RBCs, they have no mitochondria. So these RBC, they depend upon glycolysis for ATP generation. They need LDH, this lactate dehydrogenase, to convert to generate NAD+, right? To generate little amount of ATP. Other thing is that lens and cornea, both of them, they are avascular, right? So they practically have no oxygen, so they rely on anaerobic glycolysis. Another thing, WBCs, same concept, often work in hypoxic situation, often work in inflamed tissue, so they need anaerobic this glycolysis. In testes, it's really cool. What happens here is that germ cells have low O2 zones, okay? So germ cells have low O2 area, and LDH maintains this. LDH is lactate dehydrogenase. So this lactate dehydrogenase maintains this glycolysis, okay? Other thing is medulla, especially the outer medulla, because cortex gets like 90% of the blood supply, renal blood supply, and inner medulla gets like around 8%, the left 8%, and remaining 1% supply is on the outer medulla. So again, oxygen poor environment, anaerobic ATP needed. So that's the reason that lactate dehydrogenase, LDH, is found in these tissues. Okay, now physical relevance of this B3, it's called this B3, right? It's called niacin, okay? Vitamin B3 is a cofactor of LDH, and physical relevance of niacin is that niacin forms NAD+ or NADH. And now this is the main redox currency. Without NAD+ or without NADH, the glycolysis stops. So LDH keep glycolysis alive by generating it. So that's the concept. So in whole, I want to say this is an aerobic this is anaerobic condition. LDH is needed. LDH generates NAD+. Now later NAD+ allows the survival under hypoxia, okay? That's the concept. Now let's talk about second pathway. That's pyruvate turns into alanine, and this pathway is called alanine aminotransferase pathways. It means that we are transferring aminos, which in turn it means that we are just transferring carbons across tissues. So the main concept here is that we convert pyruvate into alanine with the help of ALT, and now we know that ALT is produced by liver. Now, why we need to produce alanine? Because alanine can practically jump from muscles to liver, okay? Now we need to recycle carbon between muscles to liver, but we cannot make pyruvate jump from muscles to, liver. So the enzyme here is called alanine aminotransferase, that is ALT, right? And the cofactor here is vitamin B6, and it happens in cytosol, okay? Both of these reactions happen in cytosol. And now this reaction is reversible as well. So here it goes like this. Here it goes like this. Why this happens? What's the physical relevance? In muscles, glycolysis produces pyruvate, yes, plus a little bit of ammonia. Now, why do we get ammonia? We get ammonia from the breakdown of amino acids. Now, pyruvate accepts those amino groups and forms alanine, okay? We form alanine from these amino acids, right? Now, this alanine can travel through blood and will travel to liver. Once it reaches to liver, the ALT does the reverse, alanine to pyruvate plus ammonia plus urea. Sorry, it gives us urea. So let's try to understand. Alanine, now this alanine, once is converted with ALT from ALT, and the cofactor is B6. Now this alanine in the liver goes inside this Cahill cycle. Now what happens here is this: in muscles, the pyruvate with the help of ALT turns into alanine, right? Glutamate is also one of a cofactor which gives us alpha Kg. Now later this alanine can jump from muscles to liver, right, through blood. Once it's inside the blood, this alanine turns into pyruvate again. And again, the cofactor here is alpha Kg. So essentially we are just reversing the whole reaction. And once we get the pyruvate inside the liver, we can, start gluconeogenesis, and then we can turn this pyruvate into essentially glucose. And then from glucose, we can again, this glucose can now jump to the muscles, and then it can perform something called as glycolysis. Now again, this glucose can be converted into pyruvate, in the glycolysis, back to pyruvate, becomes the pyruvate, right? That's the concept. So this whole pathway is called glucose-alanine cycle, right? And why the pyruvate can't go directly to the liver? It's a great question. The answer is that pyruvate is unstable, really unstable. It cannot cross cell membrane really easily. But on the other hand, beta-alanine is quite stable, and it carries both carbon, right? Both carbon, glucose, okay, and nitrogen for the jumping, right? Because it's more stable. Now, what is alanine? We convert pyruvate with the help of ALT to alanine. What the hell is alanine? Alanine is just an amino acid, and it is a carbon-nitrogen shuttle. That's it, okay? What is ALT here? What is glutamate here? What is alpha Kg here, okay? Now, glutamate is the main amino group carrier in this whole metabolism. And alpha Kg is called alpha ketoglutarate. And it's actually a TCA intermediate. TCA means it's this electron transport chain. What happens here is that this is one of the intermediary of this whole cycle, okay? So it's used in TCA cycle. Another question that rises here is that B6 is the cofactor. Why B6? Well, there's something called Schiff base, which is formed by B6, okay? So B6 forms something called a Schiff base. It's a base, right? It's a base with amino acid. It helps in transferring amino groups in transamination, okay? So without B6, the transaminations stop. So once the transaminations stop, without the base that is presented with the help of vitamin B6, the amino acids metabolism will fail. The physical relevance of this whole pathway is that it allows muscle to get rid of nitrogen safely, right? And it allows the muscle to recycle pyruvate carbons for glucose, right, in the liver. It's called gluconeogenesis, right? And we can, maintain the blood glucose during fasting. That's how we maintain the blood glucose during fasting, okay? Because here, as you can see, once the pyruvate is inside the liver, it can go under gluconeogenesis, and we can produce some glucose for your, glucose, right, for your blood sugar, right? So that was the second pathway. The third pathway is called oxaloacetate. Now, this oxaloacetate is actually a failsafe for gluconeogenesis, right? If we have something short on gluconeogenesis, we can use oxaloacetate, okay? Or we can use it for TCA cycle. Let's talk about it a little bit. So oxaloacetate, OAA, okay? OAA. Now, this OAA enzyme, right? The enzyme we use here to convert pyruvate into OAA is something called as pyruvate carboxylase, okay? Now, this pyruvate carboxylase is also called biotin. Cool. Now, this whole thing happens in mitochondria. And one cool thing about it is that it requires ATP plus CO2. And note that this whole thing is irreversible. There is no coming back. Okay? So why does this even happens? Well, I told you that oxaloacetate, OAA, is like a raw material for gluconeogenesis, okay, to make glucose in fasting, and it can be used for TCA cycle replenishment, right? So pyruvate carboxylase acts as a failsafe or a refiller enzyme, okay? And why B7? This is B7, by the way. Biotin is B7. So why biotin is the enzyme, right? In the main enzyme here, because biotin carries carbon dioxide, and we need carbon dioxide for this whole conversion, right? So ATP is also being used here, right? This whole pathway uses ATP to activate CO2 and attach it to pyruvate. That in turns make oxaloacetate. Why it happens in mitochondria? Well, TCA cycle and acetyl-CoA are present in the mitochondria. So these enzymes have to stay in the mitochondria, right? If acetyl-CoA builds up, a lot, the TCA cycle gets overloaded, right? So this in turn tells the cell that, hey, let's try to balance this oxaloacetate, okay? So that's the whole concept. That's why you need this whole raw materials and everything inside the mitochondria, right? The physical relevance of this whole thing is that this enzyme called pyruvate dehydrogenase complex, right? No, sorry. I'm sorry. Not this This pyruvate carboxylase, this whole enzyme, this is that it makes sure that the TCA cycle runs smoothly. It never runs out of OAA, right? It provides a good substrate for gluconeogenesis during fasting, especially in liver and kidney. Okay, so last pathway is pyruvate that turns into acetyl-CoA. Now we know that acetyl-CoA is one of the major reactant for TCA cycle. Now this pyruvate dehydrogenase, right? This is the main enzyme that plays a big role here. Now this is a complex, okay? How? Because it has this B1, B2, B3, B5, and lipoic acid. These are all 5 cofactors of pyruvate dehydrogenase. Okay, so these are like big cofactor, right? The function of these are different a little bit. For example, I can tell you about lipoic acid. This lipoic acid, it's one of the most important acid that transfers acyl group, and it plays a big role in redox reaction. Okay, so why it even have these 5 cofactors? Why? Because this pyruvate dehydrogenase PDH does 3 things. Okay, it performs something called as decarboxylation, right? That's number 1. Number 2 is oxidation. That's number 2. Number 3 does is acyl transfer. That's done by the lipoic acid, right? So all of these complex and multiple things are done by this singular, thing. So that's why it has 5 cofactors, because all of these 5 cofactors perform different shit. Okay, so I hope we understand this. I hope we understand the physical relevance of this pyruvate dehydrogenase, because pyruvate dehydrogenase actually makes acetyl-CoA, and acetyl-CoA can perform TCA. TCA cycle and fat synthesis. Note that both of the reaction that are happening in mitochondria are irreversible. Okay, and deficiency of any enzyme can actually, makes, deficiency of, for example, B1, it's called thiamine. It can cause our brain and heart failure, because again we are not able to produce TCA, and we're not able to produce ATP, loss of ATP. So the big picture here is that if it's happening in cytosol, it's reversible. If it's happening in mitochondria, it's irreversible. Another cool thing is that we use ALT here for alanine, right? And LDH for lactic acid, PDH for acetyl CoA, and biotin for oxaloacetate. Okay, so that was the whole four pathways for pyruvate.